Now let's take a look at how cytoplasm and the cytoskeleton form the cell's internal environment and provide its physical support. And not only that, it can also generate movement. If you can imagine yourself inside the cell, and imagine that cell was the size of a big lecture hall, it might come as a surprise to you that you can barely see that big rig of a nucleus parked in the front rows. Visibility is almost zero, not only because the room is filled with that jelly like cytoplasm, but also because there is a dense web of thick and thin, straight and branched ropes, strings, and scaffolding running every which way throughout the room. This inner scaffolding, which is made from proteins, is called the cytoskeleton. It has three chief purposes. You might want to write these down. First, it gives animal cells shape and support, making things like red blood cells look like the little round donuts without the hole in the middle, and giving neurons their very long thread like appearance. Plant cells are shaped primarily by their cell wall, a structure we will discuss later on, but they also have a cytoskeleton. The second chief purpose is that the cytoskeleton controls the intercellular traffic flow. Second, the cytoskeleton controls the intracellular traffic flow, serving as a series of tracks on which a variety of organelles and molecules are guided across and around the inside of the cell. And third, because the elaborate scaffolding of the cytoskeleton is dynamic and can generate force, it gives all cells some ability to control their movement. Three types of protein fibers make up the cytoskeleton. First is microtubules. It's the thickest of the three, and they are linear polymers of a protein, and they look like rigid hollow tubes. Like intracellular conveyor belts, they are the tracks to which molecules and organelles within the cell can become attached to and move along. Microtubules also help to pull chromosomes apart during cell division. Continuously built, disassembled, and rebuilt, microtubules rarely last more than about 10 minutes in a cell. You might remember seeing these in the virtual 3D tour we took of the cell a few weeks ago. Intermediate filaments are the second type of cytoskeleton fiber, and they are a durable, rope-like system of numerous different overlapping proteins. They give cells great strength. Microfilaments, the third, are the thinnest of the elements in the cytoskeleton. They're long, solid, rod-like fibers, and they help to generate forces, including those important in cell contraction and cell division. Now, a couple of microtubule-based structures are sometimes present in cells and can help to move the cell through its environment, or in stationary cells can help move the environment past the cell. Cilia are short projections often found in large numbers on a single cell. Cilia beat swiftly, often in unison and in ways that resemble blades of grass in a field blowing in the wind, or like oars of a boat, all being rowed at the same time. Cilia can move fluid along and past a cell. This movement can accomplish many important tasks, including sweeping the airways to our lungs to clear them of debris, such as dust, in the air that we're breathing. Flagella, on the other hand, are much longer than cilia. They occur in many prokaryotes and single-celled eukaryotes, as well as in many algae and plants that have cells, and they can have one or more flagella. But in animals, cell types with one or more flagella are very rare. One of these cell types, however, has a critical role in every animal species: sperm cells. With a flagellum for a tail, sperm are among the most mobile of all animal cells. Some spermicidal birth control methods prevent conception by disabling the flagellum and therefore immobilizing the sperm cells.